What's up, guys? It's your boy Dr.AnabolicCock, and this is a deep dive on the anabolic androgenic steroid testosterone. Before we start, what are anabolic steroids? Anabolic steroids act at androgen receptors to influence cellular functioning and gene expression. In addition to regulating pathways involved in the development of male characteristics, activation of androgen receptors also produces rapid increases in calcium levels within skeletal muscle, heart, and brain cells. Testosterone is a sex hormone that plays important roles in the body. In men, it's thought to regulate sex drive, libido, bone mass, fat distribution, muscle mass and strength, and the production of red blood cells and sperm. A small amount of circulating testosterone is converted to estradiol, a form of estrogen. From testosterone, we get the androgens DHT, dehydroepiandrosterone, and nandrolone. DHT is the most potent hormone among the androgens and is considered a pure androgen as it cannot convert into estrogen. It is formed primarily in the peripheral tissues of the body where it exerts its effects. Testosterone converts to DHT by the action of the 5-alpha reductase enzyme at these target tissues. Nandrolone acts as an androgen receptor agonist that is not converted endogenously to DHT, 15. As such, it provides negative feedback to the HPG axis to suppresses testosterone levels, further decreasing the available testosterone in DHT, compounding its negative effects on erectile function. Now let's get started. The king, the OG, the forefather, and the pioneer of this whole world, testosterone is the reigning champ. It's the father of all other steroids available to us today. Testosterone is what makes us men. Our whole being and makeup stem from testosterone. Along the way, someone decided that it was a good idea for us to have more. And here we are today. It adds lean muscle, helps with fat loss, raises libido, and generally amplifies our whole personality. If you're here, you should have a good idea of what it's all about. We will be treating testosterone a little differently than the rest. Below, we will go over the cost, protocol, and some general good-to-know info of a testosterone cycle. This will guide you along the way as you add other compounds and down the road. There is more than one correct way to use testosterone. But with that said, a standard testosterone cycle has been established by our gear forefathers, and would be foolish for a newbie to attempt to reinvent the wheel and deviate from the template that has been time-tested over decades by millions of men to be excellent. The standard blast is to use testosterone enanthate, or testsipionate, at 500 mgd week for 16 weeks. Hormones, relative to other drugs, are slow-acting, and it takes approximately 4-6 weeks into a fresh test enanthate cypionate blast for the drug to reach the peak effects. Gains in progress tend to be their best from about week 4 until they start to plateau around weeks 12, 16. Stopping a cycle too early at, say, 8 weeks, robs the user of the benefits of the most effective part of the cycle, and extending a cycle too long wastes the drug with diminishing returns. A 12, 16-week first-time blast of test enanthate cypionate at 500 mg week is a solid and responsible game plan to follow. A cycle of testosterone relative to other drugs and popular male hobbies is remarkably cheap. Advanced steroid stacks done by experienced full-time bodybuilders can become very expensive, but the fundamental and appropriate first-time testosterone blast pleasantly surprises men in its affordability. Testosterone is not an inferior steroid compared to more advanced and expensive steroids. Testosterone is king, and it is the hormone from which the other steroids are derived. In the United States Steroid Underground, a 10 ml vial of test enanthate, or testsipionate, at concentrations of 200 mg/ml, 250 mg/ml, or 300 mg/ml ranges in price from about $1.50 per vial to $1.100 depending on the source and middlemen. A 10 milliliters vial of test E, 250 mg/ml, contains a total of 2,500 milligrams of test E at a dosage of 500 mg/week. One vial of test E, 250, will last approximately 5 weeks. 2,500 mg/500 mg equals sign 5. For a standard 16-week blast, depending on the concentration of the testosterone, a user should anticipate needing 4 vials of test enanthate, or testsipionate. In addition to the 4 vials of test enanthate cypionate, the user will also need 1 bottle of an aromatase inhibitor, estrogen inhibitor, to use during the cycle. This bottle costs approximately $30-40. The final component a man needs to use injectable testosterone are the needles and syringes, pins. Pins are sold in 100-count boxes and cost approximately $8.30 per box depending on the source. So in total, a 16-week cycle of testosterone and post-cycle therapy will cost approximately 4 vials Test-E or CIP, $150-321 bottle of an aromatase inhibitor, $30-40 Nolvadex, $30-40 HCG, optional but strongly recommended, $30-40 pins, $25.70, total on low end, $265, total on higher end, $510. What are the benefits of blasting testosterone? Several years ago, a study was done on drug use and income levels. The researchers were curious about which recreational drug habits had the most damaging impact on a user's income. Predictably, it was shown that drug use had a negative impact on income across the board compared to those who abstained, and that included alcohol use. But there was one exception. Steroid users were found to have a higher average income than the abstaining population. This comes as no surprise to the authors because testosterone built civilizations. It makes men physically stronger, more motivated, and goal-oriented. Studies have also shown testosterone levels in men are positively correlated to cognitive function. It is common for men to report that using testosterone was life-changing. It has saved marriages, helped men make significant career advances, and of course won sporting championships. However, using testosterone for that purpose is frowned upon by the natural population. However, it is the one thing that makes baseball exciting. Anabolic steroids are a class of medications that contain a synthetically manufactured form of the hormone testosterone, or a related compound that is derived from, or similar in structure and action to, this hormone. In order to fully grasp how anabolic steroids work, it is therefore important to understand the basic functioning of testosterone. Testosterone is the primary male sex hormone. It is MANUFACTUREIDBYTHECELLSINTHESATVARYING, amounts throughout a person's lifespan. The effects of this hormone become most evident during the time of puberty, when an increased output of testosterone will elicit dramatic physiological changes in the male body. This includes the onset of secondary male characteristics such as a deepened voice, body, and facial hair growth, increased oil output by the sebaceous glands, development of sexual organs, maturation of sperm, and an increased libido. Indeed, the male reproductive system will not function properly if testosterone levels are not significant. All such effects are considered the masculinizing or androgenic properties of this hormone. Increased testosterone production will also cause growth-promoting or anabolic changes in the body, including an enhanced rate of protein synthesis, leading to muscle accumulation. Testosterone is the reason males carry more muscle mass than women, as the two sexes have vastly contrasting amounts of this hormone. More specifically, the adult male body will manufacture between 2.5 and 11 mg per day 1, while females only produce about 1/4 mg. The dominant sex hormone for women is estrogen, which has a significantly different effect on the body. Among other things, a lower androgen and higher estrogen level will cause women to store more body fat, accumulate less muscle tissue, have a shorter stature, and become more apt to bone weakening with age. The testosterone molecule will become bound to the intracellular receptor site, located in the cytosol, not on the membrane surface, forming a new receptor complex. This complex, hormone plus receptor site, will then migrate to the cell's nucleus, where it will attach to a specific section of the cell's DNA, referred to as the hormone response element. This will activate the transcription of specific genes, which in the case of a skeletal muscle cell will ultimately cause, among other things, an increase in the synthesis of the two primary contractile proteins, actin and myosin, muscular growth. Carbohydrate storage in muscle tissue may be increased due to androgen action as well. Once this messaging process is completed, the complex will be released and the receptor and hormone will disassociate. Both are then free to migrate back into the cytosol for further activity. The testosterone molecule is also free to diffuse back into circulation to interact with other cells. The entire receptor cycle, including hormone binding, receptor hormone complex migration, gene transcription, and subsequent return to cytosol, is a slow process taking hours, not minutes, to complete. For example, in studies using a single injection of nandrolone, it is measured to be 4 to 6 hours before free androgen receptors migrate back to the cytosol after activation. It is also suggested that this cycle includes the splitting and formation of new androgen receptors once returned to cytosol, a possible explanation for the many observations that androgens are integral in the formation of their own receptor sites. 2. In the kidneys, this same process works to allow androgens to augment erythropoiesis, red blood cell production. Point 3. It is this effect that leads to an increase in red blood cell concentrations and possibly increased oxygen transport capacity during anabolic androgenic steroid therapy. Many athletes mistakenly assume that oxymethylone and boldenone are unique in this ability due to specific uses or mentions of this effect in drug literature. In fact, stimulation of erythropoiesis occurs with nearly all anabolic androgenic steroids, as this effect is simply tied with activation of the androgen receptor in kidney cells. The only real exceptions might be compounds such as dihydrotestosterone and some of its derivatives. 4. Which are rapidly broken down upon interaction with the 3 alpha-hydroxysteroid dehydrogenase enzymes. Kidney tissue has a similar enzyme distribution to muscle tissue. See anabolic/androgen dissociation section, and therefore display low activity in these tissues. Adipose, fat tissues are also androgen responsive, and here these hormones support the lipolytic, fat mobilizing capacity of cells. This may be accomplished by an androgen-tied regulation of beta-adrenergic receptor concentrations or general cellular activity through adenylate cyclase. Point 6. We also note that the level of androgens in the body will closely correlate inversely with the level of stored body fat. As the level of androgenic hormones drops, typically the deposition of body fat will increase. 7. Likewise, as we enhance the androgen level, body fat may be depleted at a more active rate. The ratio of androgen to estrogen action is in fact most important, as estrogen plays a counter role by acting to increase the storage of body fat in many sites of action. Likewise, if one wished to lose fat during steroid use, estrogen levels should be kept low. This is clearly evidenced by the fact that non-aromatizing steroids have always been favored by bodybuilders looking to increase the look of definition and muscularity, while aromatizing compounds are typically relegated to bulking phases of training due to their tendency to increase body fat storage. Aromatization is discussed in more detail in a following section. See estrogen aromatization. As mentioned, testosterone also elicits androgenic activity, which occurs by its activating receptors in what are considered to be androgen-responsive tissues, often through prior conversion to dihydrotestosterone. See DHT conversion. This includes the sebaceous glands, which are responsible for the secretion of oils in the skin. As the androgen level rises, so does the release of oils. As oil output increases, so does the chance for pores becoming clogged. We can see why acne is such a common side effect of steroid use. The production of body and facial hair is also linked to androgen receptor activation in skin and scalp tissues. This becomes most noticeable as boys mature into puberty, a period when testosterone levels rise rapidly and androgen activity begins to stimulate the growth of hair on the body and face. Sometime later in life, and with the contribution of a genetic predisposition, androgen activity in the scalp may also help to initiate male pattern hair loss. It is a misconception that dihydrotestosterone is an isolated culprit in the promotion of hair loss, however, as in actuality it is the general activation of the androgen receptor that is to blame. See DHT conversion. The functioning of sex glands and libido are also tied to the activity of androgens, as are numerous other regions of the central nervous/neuromuscular system. Although testosterone has been isolated, synthesized, and actively experimented with for many decades now, there is still some debate today as to exactly how steroids affect muscle mass. At this point in time, the primary mode of anabolic action with all anabolic androgenic steroids is understood to be direct activation of the cellular androgen receptor and increases in protein synthesis. As follows: If we are able to increase our androgen level from an external source by supplementing testosterone or a similar anabolic steroid, we can greatly enhance the rate in which protein is retained by the muscles. This is clearly the primary cause for muscle growth with all anabolic androgenic steroids. As our hormone levels increase, so does androgen receptor activation, and ultimately the rate of protein synthesis. But other indirect mechanisms could possibly affect muscle growth outside of the normally understood androgen action on protein synthesis. An indirect mechanism is one that is not brought about by activation of the androgen receptor, but the affect androgens might have on other hormones, or even the release of locally acting hormones or growth promoters inside cells, perhaps mediated by other membrane-bound receptors. We must remember also that muscle mass disposition involves not only protein synthesis, but also other factors such as tissue nutrient transport and protein breakdown. We need to look at androgenic interaction with these factors as well to get a complete picture. Concerning the first possibility, we note that studies with testosterone suggest that this hormone does not increase tissue amino acid transport. This fact probably explains the profound synergy bodybuilders have noted in recent years with insulin, a hormone that strongly increases transport of nutrients into muscle cells. But regarding protein breakdown, we do see a second important pathway in which androgens might affect muscle growth. Anti-glucocorticoid cold meat of testosterone, and synthetic anabolic androgenic steroids may help to increase mass and strength by having an anti-catabolic effect on muscle cells. Considered one of the most important indirect mechanisms of androgen action, these hormones are shown to affect the actions of another type of steroid hormone in the body. Glucocorticoids, cortisol is the primary representative of this group. Glucocorticoid hormones actually have the exact opposite effect on the muscle cell than androgens, namely sending an order to release stored protein. This process is referred to as catabolism and represents a breaking down of muscle tissue. Muscle growth is achieved when the anabolic effects of testosterone are more pronounced overall than the degenerative effects of cortisol. With intense training and a proper diet, the body will typically store more protein than it removes, but this underlying battle is always constant. When administering anabolic steroids, however, a much higher androgen level can place glucocorticoids at a notable disadvantage. With their effect reduced, fewer cells will be given a message to release protein, and more will be accumulated in the long run. The primary mechanism believed to bring this effect out is androgen displacement of glucocorticoids bound to the glucocorticoid receptor. In fact, in vitro studies have supported this notion by demonstrating that testosterone has a very high affinity for this receptor, 11, and further suggesting that some of its anabolic activity is directly mediated through this action. 12. It is also suggested that androgens may indirectly interfere with DNA binding to the glucocorticoid response element. 13. Although the exact underlying mechanism is still in debate, what is clear is that steroid administration inhibits protein breakdown, even in the fasted state, which seems clearly indicative of an anti-catabolic effect. Testosterone and creatinine. In addition to protein synthesis, a rise in androgen levels should also enhance the synthesis of creatine in skeletal muscle tissues. 14. Creatine, as creatine phosphate, CP, plays a crucial role in the manufacture of ATP, adenosine triphosphate, which is a main store of energy for the muscles. As the mu.scale cells are stimulated to contract, ATP molecules are broken down into ADP, adenosine diphosphate, which releases energy. The cells will then undergo a process using creatine phosphate to rapidly restore ADP to its original structure in order to replenish ATP concentrations. During periods of intense activity, however, this process will not be fast enough to compensate and ATP levels will become lowered. This will cause the muscles to become fatigued and less able to effort a strenuous contraction. With increased levels of CP available to the cells, ATP is replenished at an enhanced rate and the muscle is both stronger and more enduring. This effect will account for some portion of the early strength increases seen during steroid therapy, although perhaps not technically considered an anabolic effect, as tissue hypertrophy is not a direct result. Androgen support of creatine synthesis is certainly still looked at as a positive and growth-supporting result in the mind of the bodybuilder, testosterone and IGF-1. It has also been suggested that there is an indirect mechanism of testosterone action on muscle mass mediated by insulin-like growth factor. To be more specific, studies note a clear link between androgens and tissue release of and responsiveness to this anabolic hormone. For example, it has been demonstrated that increases in IGF-1 receptor concentrations in skeletal muscle are noted when elderly men are given replacement doses of testosterone. In essence, the cells are becoming primed for the actions of IGF-1 by testosterone. Alternately, we see marked decreases in IGF-1 receptor protein levels with androgen deficiency in young men. It also appears that androgens are necessary for the local production and function of IGF-1 in skeletal muscle cells, independent of circulating growth hormone and IGF-1 levels. Since we do know for certain that IGF-1 is at least a minor anabolic hormone in muscle tissue, it seems reasonable to conclude that this factor, at least at some level, is involved in the muscle growth noted with steroid therapy. Direct and indirect steroids? In looking over the proposed indirect effects of testosterone and pondering the effectiveness of the synthetic anabolic/androgenic steroids, we must resist the temptation to believe we can categorize steroids as those which directly and those which indirectly promote muscle growth. The belief that there are two dichotomous groups or classes of steroids ignores the fact that all commercial steroids promote not only muscle growth, but also androgenic effects. There is no complete separation of these traits at this time, making clear that all activate the cellular androgen receptor. I believe the theory behind direct and indirect steroid classifications originated when some noted the low receptor binding affinity of seemingly strong anabolic steroids like oxymethylone and methandrostenolone. If they bind poorly, yet work well, something else must be at work. This type of thinking fails to recognize other factors in the potency of these compounds, such as their long half-lives, estrogenic activity, and weak interaction with restrictive binding proteins. See, free versus bound testosterone. While there may possibly be differences in the way various compounds could foster growth indirectly, such that advantages might even be found with certain synergistic drug combinations, the primary mode of action with all of these compounds is the androgen receptor. The notion that steroid X and Y must never be stacked together because they both compete for the same receptor when stimulating growth, while X and Z should be combined because they work via different mechanisms, should likewise not be taken too seriously. Such classifications are based on speculation only, and upon reasonable investigation are clearly invalid. A very small amount of testosterone actually exists in a free state, where interaction with cellular receptors is possible. The majority will be bound to the proteins SHBG, sex hormone binding globulin, also referred to as sex steroid binding globulin, and testosterone estradiol binding globulin, and albumin, which temporarily prevent the hormone from exerting activity. Steroid hormones actually bind much more avidly to SHBG than albumin, with approximately 1,000 times greater affinity. However, albumin is present in a level 1,000 times greater than SHBG. Therefore, the activity of both B and DING proteins in the body is relatively equal. The distribution of testosterone in men is typically 45% of testosterone bound to SHBG and about 53% bound to albumin. The remaining 2% of the average blood concentration exists in a free, unbound state. In women, the percentage of free testosterone is lower, measured to be approximately 1%. A binding protein called ABP, androgen binding protein, also helps to mediate androgen activity in the reproductive system, although since it is found exclusively in these tissues, it is not relevant to muscle growth. The level of free testosterone available in the blood is likewise an important factor mediating its activity, as only a small percentage is really active at any given time. It must also be noted that as we alter testosterone to form new anabolic/androgenic steroids, we also typically alter the affinity in which the steroid will bind to plasma proteins. This is an important consideration, as the higher percentage we have of free hormone, the more active the compound should be on a milligram for milligram basis, and the variance can be substantial between different compounds. For example, proviron, 1-methyl dihydrotestosterone, binds with SHBG many times more avidly than testosterone, while mebelarone, 7,17-dimethylnandrolone, and belastirone, 7,17-dimethyltestosterone, show virtually no affinity for this protein at all. Clearly the reason these steroids are such potent androgens. The level of SHBG present in the body is also variable, and can be altered by a number of factors. The most prominent seems to be the concentration of estrogen and thyroid hormones present in the blood. We generally see a reduction in the amount of this plasma-binding protein as estrogen and thyroid content decreases, and a rise in SHBG as they increase. A heightened androgen level due to the administration of anabolic androgenic steroids has also been shown to lower levels of this protein considerably. This is clearly supported by a 1989 German study, which noted a strong tendency for SHBG reduction with the oral anabolic steroid stanozolol, Winstrol. Estrogen aromatization. Testosterone is the primary substrate used in the male body for the synthesis of estrogen, estradiol, the principal female sex hormone. Although the presence of estrogen may seem quite unusual in men, it is structurally very similar to testosterone. With a slight alteration by the enzyme aromatase, estrogen is produced in the male body. Aromatase activity occurs in various regions of the male body, including adipose, 22 liver, 23 gonadal, 24 central nervous system, 25 and skeletal muscle, 26 tissues. In the context of the average healthy male, the amount of estrogen produced is generally not very significant to one's body disposition and may even be beneficial in terms of cholesterol values, see side effects, cardiovascular disease. However, in larger amounts, it does have potential to cause many unwanted effects, including water retention, female breast tissue development, gynecomastia, and body fat accumulation. For these reasons, many focus on minimizing the buildup or activity of estrogen in the body with aromatase inhibitors such as Arimidex and Cytodren, or anti-estrogens such as Clomid or Nolvadex, particularly at times when gynecomastia is a worry, or the athlete is attempting to increase muscle definition. We must, however, not be led into thinking that estrogen serves no benefit. It is actually a desirable hormone in many regards. Athletes have known for years that estrogenic steroids are the best mass builders, but it is only recently that we are finally coming to understand the underlying mechanisms why. It appears that reasons go beyond the simple size, weight, and strength increases that one would attribute to estrogen-related water retention, with this hormone actually having a direct effect on the process of anabolism. This is manifest through increases in glucose utilization, growth hormone secretion, and androgen receptor proliferation. Glucose utilization and estrogen may play a very important role in the promotion of an anabolic state by affecting glucose utilization in muscle tissue. This occurs via an altering of the level of available glucose 6-phosphate dehydrogenase, an enzyme directly tied to the use of glucose for muscle tissue growth and recuperation. 2728. More specifically, G6PD is a vital part of the pentose phosphate pathway, which is integral in determining the rate nucleic acids and lipids are to be synthesized in cells for tissue repair. During the period of regeneration after skeletal muscle damage, levels of G6PD are shown to rise dramatically, which is believed to represent a mechanism for the body to enhance recovery when needed. Surprisingly, we find that estrogen is directly tied to the level of G6PD that is to be made available to cells in this recovery window. The link between estrogen and G6PD was established in a study demonstrating levels of this dehydrogenase enzyme to rise after administration of testosterone propionate. The investigation further showed that the aromatization of testosterone to estradiol was directly responsible for this increase and not the androgenic action of this steroid. The non-aromatizable steroids dihydrotestosterone and fluoxymesterone were tested alongside testosterone propionate, but failed to duplicate the effect of testosterone. Furthermore, the positive effect of testosterone propionate was blocked when the aromatase inhibitor 4-hydroxyandrostenedione, formastane, was added, while 17-beta estradiol administration alone caused a similar increase in G6PD to testosterone propionate. The inactive estrogen isomer 17, alpha estradiol, which is unable to bind the estrogen receptor, failed to do anything. Further tests using testosterone propionate and the anti-androgen flutamide showed that this drug also did nothing to block the positive action of testosterone, establishing it as an effect independent of the androgen receptor. Estrogen and GHIGF-1 estrogen may also play an important role in the production of growth hormone and IGF-1. IGF-1, insulin-like growth factor, is an anabolic hormone released in the liver and various peripheral tissues via the stimulus of growth hormone. See drug profiles, growth hormone. IGF-1 is responsible for the anabolic activity of growth hormone, such as increased nitrogen retention, protein synthesis, and cell hyperplasia, proliferation. One of the first studies to bring this issue to our attention looked at the effects of the anti-estrogen tamoxifen on IGF-1 levels, demonstrating it to have a suppressive effect. A second, perhaps more noteworthy study took place in 1993, which looked at the effects of testosterone replacement therapy on GH and IGF-1 levels alone, and compared them to the effects of testosterone combined again with tamoxifen. When tamoxifen was given, GH and IGF-1 levels were notably suppressed, while both values were elevated with the administration of testosterone enanthate alone. Another study has shown 300 milligrams of testosterone enanthate weekly to cause a slight IGF-1 increase in normal men. Here, the 300 milligrams of testosterone ester caused an elevation of estradiol levels, which would be expected at such a dose. This was compared to the effect of the same dosage of nandrolone decanoate. However, this steroid failed to produce the same increase. This result is quite interesting, especially when we note that estrogen levels were actually lowered when this steroid was given. Yet another demonstrated that GH and IGF-1 secretion is increased with testosterone administration on males with delayed puberty, while dihydrotestosterone, non-aromatizable, seems to suppress GH and IGF-1 secretion, estrogen, and the androgen receptor. It has also been demonstrated that estrogen can increase the concentration of androgen receptors in certain tissues. This was shown in studies with rats, which looked at the effects of estrogen on cellular androgen receptors in animals that underwent orchiectomy, removal of testes, often done to diminish endogenous androgen production. According to the study, administration of estrogen resulted in a striking 480% increase in methyltrianolone, a potent oral androgen often used to reference receptor binding in studies, binding in the levator ANI muscle. The suggested explanation is that estrogen must either be directly stimulating androgen receptor production or perhaps diminishing the rate of receptor breakdown. Although the growth of the levator ANI muscle is commonly used as a reference for the anabolic activity of steroid compounds, it is admittedly a sex organ muscle and different from skeletal muscle tissue in that it possesses a much higher concentration of androgen receptors. This study, however, did look at the effect of estrogen in fast-twitch skeletal muscle tissues, tibialis anterior and extensor digitorum longus, as well, but did not note the same increase as the levator ANI. Although discouraging at first glance, the fact that estrogen can increase androgen receptor binding in any tissue remains an extremely significant finding, especially in light of the fact that we now know androgens to have some positive effects on muscle growth that are med-iated outside OF muscle tissue. Estrogen and fatigue. Steroid fatigue is a common catchphrase these days and refers to another important function of estrogen in both the male and female body, namely its ability to promote wakefulness and a mentally alert state. Given the common availability of potent third-generation aromatase inhibitors, bodybuilders today are, at times, noticing more extreme estrogen suppression than they had in the past. Often associated with this suppression is fatigue. Under such conditions, the athlete, though on a productive cycle of drugs, may not be able to maximize his or her gains due to an inability to train at full vigor. This effect is sometimes also dubbed steroid lethargy. The reason is that estrogen plays an important supporting role in the activity of serotonin. Serotonin is one of the body's principal neurotransmitters, vital to mental alertness and the sleep-wake cycle. Interference with this neurotransmitter is also associated with chronic fatigue syndrome, so we can see how vital it is to fatigue specifically. Estrogen suppression in menopause has also been associated with fatigue, as has the clinical use of newer, more potent aromatase inhibitors like anastrozole, letrozole, exemestane, and fedrozole in some patients. These things may be important to consider when planning your next cycle. Although not everyone notices this problem when estrogen is low, for those that do, a little testosterone or estrogen can go a long way in correcting this. It is also of note that the use of strictly non-aromatizable steroids sometimes causes this effect as well, likely due to the suppression of natural testosterone production, cutting off the main substrate used by the male body to make estrogen. Anti-estrogens and the athlete. So what does this all mean to the bodybuilder looking to gain optimal size? Basically, I think it calls for a cautious approach to the use of estrogen maintenance drugs if mass is the key objective. Things change, of course, if we are talking about cutting. Obviously, anti-estrogens should be used if there is a clear need for them due to the onset of estrogenic side effects, or at the very least, the drugs being administered should be substituted for non-estrogenic compounds. Gynecomastia is certainly an unwanted problem for the steroid user, as are noticeable fat mass gains. But if these problems have not presented themselves, the added estrogen due to a cycle of testosterone or dianabol, for example, might indeed be aiding in the buildup of muscle mass, or keeping you energetic. An individual confident they will notice, or are not prone to getting, estrogenic side effects may therefore want to hold off, using estrogen maintenance drugs so as to achieve the maximum possible gains in tissue mass, DHT conversion. As we see from our discussion with estrogen and considering the physiological effects of any steroid, we must look at all of its active metabolites and not just the initial compound. This includes not only estrogenic products, but androgenic metabolites as well. With this in mind, it is important to note that the potency of testosterone is considerably increased in many androgen-responsive tissues when it converts to dihydrotestosterone. More commonly referred to by the three-letter abbreviation DHT, this hormone is in fact measured to be approximately 3 to 4 times stronger than testosterone. It is the most potent steroid found naturally in the human body, and important to discuss if we are to understand the full activity of testosterone, as well as other anabolic androgenic steroids that undergo a similar conversion. Testosterone is converted to dihydrotestosterone upon interaction with the 5-alpha reductase enzyme. More specifically, this enzyme removes the C4-5 double bond of testosterone by the addition of 2 hydrogen atoms to its structure, hence the name dihydrotestosterone. The removal of this bond is important, as in this case, it creates a steroid that binds to the androgen receptor much more avidly than its parent steroid. 5-alpha reductase is present in high amounts in tissues of the prostate, skin, scalp, liver, and various regions of the central nervous system, and as such represents a mechanism for the body to increase the potency of testosterone, specifically where strong androgenic action is needed. In these areas of the body, little testosterone will actually make its way to the receptor without being converted to dihydrotestosterone, making DHT by far the active form of androgen here. DHT and androgenic side effects. In some regards, this local potentiation of testosterone's activity may be unwelcome, as higher androgenic activity in certain tissues may produce a number of undesirable side effects. Acne, for example, is often triggered by dihydrotestosterone activity in the sebaceous glands, and the local formation of dihydrotestosterone in the scalp is typically blamed for triggering male pattern hair loss. You should know that it is a terrible misconception among bodybuilders that dihydrotestosterone is an isolated culprit when it comes to these side effects. All anabolic androgenic steroids exert their activities, both anabolic and androgenic, through the same cellular androgen receptor. Dihydrotestosterone is no different than any other steroid, except that it is a more potent activator of this receptor than most, and can be formed locally in certain androgen-sensitive tissues. All steroids can cause androgenic side effects in direct relation to their affinity for this receptor, and DHT has no known unique ability in this regard. Benefits of DHT. While a lot of attention is being paid to the negative side effects of the androgen dihydrotestosterone, you should know that there are some known benefits to the strong androgenic activity brought about by this hormone as well. For example, DHT plays an important role in the organization and functioning of the central nervous system. Many neural cells contain active androgen receptors, and it is thought that there may even be a specific importance of dihydrotestosterone in this area of the body. Studies have shown DHT to have a profoundly greater impact in these cells compared to testosterone. More specifically, animal models demonstrated that both testosterone and DHT would result in increased androgen receptor proliferation in neural cells 3 and 7 hours after being administered. However, only DHT was able to sustain this increase at the 21-hour mark. Although some might contend that this difference is simply due to DHT forming a more stable and lasting complex with the androgen receptor, others suggest that DHT and testosterone might even be affecting neural cells differently, such that the dihydrotestosterone receptor complex and testosterone receptor complex might be activating the transcription of different target genes. The strong interaction between the central nervous system and skeletal muscles, collectively referred to as the neuromuscular system, is of key importance to the athlete. There appears to be little doubt that the ability of the body to adapt to training and to activate nerve endings in muscle tied at SU is reliant on the interactions of the neuromuscular system. Inhibiting the formation of DHT during a testosterone cycle may therefore inadvertently interfere with strength and muscle mass gains. This would explain why bodybuilders commonly report a drop in steroid potency when they add the S-alpha reductase inhibitor finasteride to a testosterone cycle. Many complain strength and even muscle mass gains slow significantly when this medication is added, which would not make sense if testosterone and androgen receptor activation in muscle tissue were solely responsible for growth. Clearly, more is involved, and we cannot look at dihydrotestosterone simply as a side effect hormone. Alright guys, that's it for this deep dive, the anabolic androgenic steroid testosterone, and I hope you enjoyed and learned something from this video. Dr. AnabolicCock out.